And I'll just jump in now. Is that all
Right.
right? All
So we
right.
want to welcome you, Dr. Bailey, and we owe you one for agreeing to introduce yourself so that no errors are made. So without further ado, please begin anytime.
Wonderful. Thanks. Yeah, I'm very excited to be here. My name is Jordan Bailey. I am a toxicology specialist in the Bureau of Environmental Health at the State Health Department. That's a brand new bureau for those keeping track. We used to be the Division of Environmental Health, and sort of excited to expand into that bureau space. But yeah, I serve largely a role in our PFAS health research. So I'm principal investigator, study director on several different PFAS health studies that I'm going to talk to you all about today. My background is much more in neurotoxicology. So my doctoral and postdoctoral training were both within that sort of subfield of neurotox and behavioral effects of exposure to contaminants from our environment. And since coming to the state health department, I really moved into more of this landscape of studying humans instead of animal models and doing some really important public health investigative work. So that's what I'll dive into now. And I think I have only 40-ish, 45-ish minutes planned. So I think that'll leave plenty of time for questions at the end. And I'll be very excited to chat with y'all about any of this stuff. And I may even end a little earlier than that, which is great because I'll welcome any questions or comments. Okay, but with that, I'll just dive in. So the main thing I want to talk about is the ongoing research that's been happening at DHHS, including looking back just a little bit in time at where some of this started. So I'm going to focus really on PFAS exposures, like our understanding of PFAS exposures, our understanding of PFAS blood levels, and their associated health effects. And so I really want to share, or at least start by sharing, the general landscape of the work that we're doing at the Michigan Department of Health and Human Services on PFAS. That spans from exposure assessments to cross-sectional and longitudinal epidemiological studies, all the way up to statewide biomonitoring and surveillance activities. But of all of that, the work that I'll focus on today is just a couple examples. So I'll do a deeper dive into just really two of these. But I'm always willing to come back and talk more about some of the other exciting work that we're doing. But to give you an understanding of what I'm presenting on the screen here, these are just our study icons, but I'm going to talk through them a little bit. What I have in green outline, so you can, I don't know if you see my cursor, but the green outline ones are our exposure assessments in PFAS. So we have the North Kent County exposure assessment, and we have the Oscoda area exposure assessment. So these are projects where MDHHS goes into a community that has experienced environmental contamination with PFAS and where there are questions about what that exposure may mean to people. And to date, we have been able to set up two such exposure assessments in these two key places. So one is where there was really extreme high levels of PFAS found in drinking water. That's the North Kent County Exposure Assessment one, and I'll talk much more about that one in just a moment. And then the other one is where there were a lot of questions about the various routes of exposure to PFAS, like wild game, especially deer, and fish, and some other sources that go beyond drinking water, beyond questions of exposure via drinking water. And that's the Oscoda Area Exposure Assessment OAEA one here in green. And I'm not really going to focus on that one today. But in the dark blue outlines, see here, these are our like proper epidemiological studies. So this is the Michigan PFAS Exposure and Health Study. We refer to it as MIPES. The multi-site study, also referred to as MSS. We have BALANCE, that's behavioral adaptability, learning about novel contamination in the environment. And PAIR, right under that, that's our PFAS exposure and antibody response to COVID-19 vaccine. And finally, EMBARK, way over here at the bottom. EMBARK is the Environmental Mediators of Birth Effects in Relation to Contaminants study. And so all of these in the dark blue outline are really large-scale public health investigations where we're specifically interested in advancing generalizable knowledge about the health effects of PFAS. And of all of these, I'm really going to focus on MIPES today and a little bit more, I'll give a little bit more context on multi-site study, MSS, because those are really considered our flagship PFAS health studies. These are really high-profile ones. We're engaging rigorous research here, and they're designed to understand how PFAS blood levels really do relate to health. But I will give you at least a basic picture of what balance, pair, and embark are as well. Embark is brand new, and so I'll mention just a few words about that in a moment. Okay, but to round out this landscape, the light blue outlines. So those are the ones here and here. Those are the ones that are surveillance or biomonitoring in nature. So that's the Michigan Chemical Exposure Monitoring Program and the PFAS and Firefighters of Michigan Surveillance Program. And I'm actually really not going to go into those much at all. I'm really going to just note that those are large-scale biomonitoring programs. They aim to either establish a statewide reference value for PFAS in blood, that's my chem, or they aim to characterize PFAS exposures sort of to an entire job sector, and that's PFOAMs. So combined, everything on your screen that you see here, like all of these projects involve thousands of participants, like many, many data points, and they're just now beginning to produce some impactful results with findings that should advance not only the scientific literature, but also inform public health policy, hopefully for years to come. And the way I've arranged these on the screen here for you is generally, loosely, I would say, by chronological order, from the earliest up here with North Kent County Exposure Assessment down to the most recent, which is Embark. And that's sort of how I want to tell the story today. I just want to start with where all of this work largely originated and then take you through time as our scope has sort of expanded and adapted to the environment. So let's start with that very first exposure assessment, the one called North Kent County Exposure Assessment. That took place on the west side of the state in an area impacted by very high levels of environmental contamination of PFAS. And so it is generally understood that PFAS in this area resulted from PFAS containing manufacturing waste that leached from unlined landfills. Those landfills were dispersed across a wide geographic area. And as you can see here in this map, the state's environmental investigation revealed pockets of extremely contaminated groundwater that was used as drinking water. The highest was over 50,000 parts per trillion in private well water. And for comparison, the current limits are in the single digits for most of the PFAS here that were found. But not only were there those pockets of really high concentrations, there were also pockets of moderate and then even no contamination found during the environmental investigation. And I'll note that this environmental investigation began way back in 2017, and it actually continues to this day. So EGLE, our sister agency, the environmental agency, is still on the ground conducting investigative work here, and the health department is still on the ground here, making sure people are protected from the threats associated with PFAS in their drinking water. So that investigation was really a multi-agency effort. It certainly involved more than just the state health department. But as you can see here, it's widespread. The contamination was widespread and it was kind of diverse. There were pockets of, like I said, really high contamination, and those folks were all moved immediately to alternate sources of drinking water. And I can say that actually for this entire area. So as this contamination was discovered, all affected residents were moved to alternate sources of drinking water, and that ranged from whole house filtration systems to expansions of municipal water systems and other interventions as well. And all of that work occurred immediately upon discovery of contamination in this area. So that was back in 2017. But in 2018, DHHS, the state health department, attempted to start answering questions brought by this community who wanted to know how this drinking water contamination had impacted them. So to do that, we launched the North Kent County Exposure Assessment, which took samples of blood from those impacted residents, and we tested it for many different PFAS, including the ones that were found at high levels in their drinking water. So the key question that we wanted to answer with that was, has this environmental contamination led to detectable concentrations of PFAS in the blood of these residents? And if so, what is that distribution of blood levels here? And what, if any, demographic or lifestyle factors are associated with those blood levels?
Could I ask a question?
Oh, yeah, for sure.
So the map you have there, the red and the yellow dots do come right up to the edge of the test area. So has additional testing being done outside that area? And more generally, what would we expect to find in just in general through the state?
Yeah. So this study area that I'm putting on the screen is the like kind of original one back when this exposure assessment was in place. It has expanded since then. So today's boundaries are wider because Eagle continue to test residential wells. There's now a full-on expansion in the Cortland Township area, which is to the east of this study map that you see here. As a direct result of more testing and expanding that, trying to find where the contamination boundaries are. So yeah, that is absolutely ongoing. And I think what the geologists and what the experts at Eagle, I think, are looking for is that that distribution from the high strength source out to like non-detects and trying to define boundaries based on those environmental data. So yes, that work is ongoing, and we've adapted. So, and I'll get to this a little bit later, we've adapted our eligibility criteria for who can be enrolled in some of these health studies, not based on the historic outlines of where, investigations started, but where they currently are. So people that are newly sampled by Eagle would continue to have.
So are you seeing the high detects and detects but below action level in the wider area that's being evaluated?
Yeah, so as Eagle continues to go out from the high strength sources, they're seeing a decline in the concentrations, as I think you would expect based on the movement of groundwater. But that and like the conceptual site model for like the environmental contamination is a bit beyond like what I work on directly, and it's beyond what like the health department is leading on. So that really is Eagle. And so I'm not going to have like really super nuanced answers to some of those important questions. But yeah, that conceptual site model is like the key thing that they're trying to understand at Eagle.
Okay, thank you.
Yeah, no, thank you. Okay, so here I wanted to note who joined North Kent County Exposure Assessment. So the eligible people who joined in provided blood samples, and they answered a lengthy exposure sources survey. So that asked them all kinds of questions about their job, their hobbies, the food that they eat, all that kind of stuff. And the demographic characteristics of those who joined are shown here. So you can see in general, people had an average age of 44 years old. We were roughly evenly distributed between males and females, 47 to 52% male to female. And you can see, and this was pretty well expected based on the actual study area that was eligible, but we did have like relatively higher income population with relatively more years of education compared to other areas in the state, and then other areas that we've conducted some of this work in. And when we look at their blood levels, and when we compare those blood levels to blood levels reported by NHANES, we can see that those are the results I'm showing you here. And so depending on the PFAS studied, North Kent County exposure area participants were either higher, lower, or about the same as compared to the national average as estimated from NHANES data. But importantly, what I want to point out is that the PFAS for which participants were higher than NHANES are the ones that were found in the highest concentrations in their drinking water. So PFOS and PFOA in particular. And those are underlined here as well. When we looked to see if any participant characteristics predicted these blood levels, the results were largely unsurprising. So age increased PFAS level, and that was of course predicted based on what we know about the half-lives of these PFAS. And males had more PFAS in general than females. Again, that was predictable based on the enhanced excretion that menstruating women have compared to males. And also unsurprising was the finding that the amount of water consumed, of drinking water consumed in the area and the years spent living in these impacted neighborhoods also predicted these PFAS serum concentrations. So nothing too surprising there, but confirmation that that environmental contamination was relevant and impactful for the blood levels of residents living in those areas. We also looked at at sort of other factors and weren't terribly surprised, again, to find that eating fish, which are known to bioaccumulate PFOS in particular, and eating chicken eggs from within the contaminated neighborhoods was also associated with PFAS serum levels. And I'll note that both kidney disease and blood donations were also associated with less PFAS in blood. Now, kidney disease has this effect, presumably because these participants were patients who are on dialysis, although we do not have rich data on that. And so that's kind of a presumption at the moment, but much more easily understood would be blood donation. That makes sense. We believe that has that effect because of the enhanced excretion then of PFAS through that blood donation route. So yeah, I'm gonna stay on the slide, but note a couple other things. Like this study, North Kent County Exposure Assessment, was really important for a number of reasons. One, we were able to directly answer the questions that these residents had. The impacted residents were asking questions about what this industrial contamination of their drinking water meant to them. And so that question was really directly, is there PFAS in my blood? And if so, how much? And we were able to provide direct answers to that question through the North Kent County Exposure Assessment. All of the participants in this received written and, when needed, verbal explanation of their PFAS blood test results. They also got extra health education geared towards understanding those results. And then they were also provided information about how to speak with their doctor about their results. But the other sort of important part here is that we were able to start to understand or disentangle some of the lifestyle or other characteristics that are associated with elevated PFAS blood levels in this community. And that really provided some nuance and texture to these overall findings, which in general were not surprising. I'm not suggesting that it was surprising that we found PFAS in blood when it was at such high levels in the water. We expected to find that, but the fact is we were able to systematically document it and also note what characteristics were associated with that finding. Okay, so following this, many of the participants in North Kent County asked for a health study. Their next question was, now that I know that this is in my blood for sure, how is it going to affect my health? And so DHHS has taken that question very seriously. And in the years following North Kent County Exposure Assessment, we have secured external federal funding for a cross-sectional PFAS health study and for other studies that are funded by external sources. And we've also secured state funding that supports a longitudinal health study. And overall, we've really been able to have this diverse landscape of health studies, all of which are aimed at better understanding PFAS's effect on health. And so that's really what has come after North Kent County. Okay. So I really want to take a moment to just talk about those in general, and then I'm going to do a deeper dive into one of them. The one I'm going to do a deeper dive in is MIPES, Michigan PFAS Exposure and Health Study. But let me start with the multi-site study. This, or MSS, is a nationwide cross-sectional study, and this is run by the Agency for Toxic Substances and Disease Registry, which was historically been within the CDC. This study provides information to communities about the health effects of exposure to PFAS. That's the stated goal of it. It all started in 2019, and it was a competitive funding opportunity from the CDC and ATSCR, and then they announced partnerships with 7 study teams who had applied for this award to carry out multi-site study locally in their communities across the nation. So DHHS was one of those selected 7 study teams. And we joined mostly academic researchers from around the country who were launching multi-site study locally. So yeah, there's 7 sites across the country that are part of multi-site study, and each of us had documented PFAS contamination of our drinking water. And that was a key part of the criterion for being eligible for that award. But my point is that data collection for the multi-site study is combined to create a nationwide data set. So there aren't really Michigan specific analyses planned as part of multi-site study proper. The entire data set is considered a whole. And that combined data set hopefully will allow us to better explore health outcomes from PFAS exposure and look at variables that wouldn't be possible if we were restricted to just the MIPES cohort. So, or excuse me, just the Michigan cohort. So in Michigan, for that award, we proposed the same Belmont Rockford area that I spoke to you about just a moment ago for the North Kent County Exposure Assessment, But to that, we added another area, which you may have heard of because there was a lot of news coverage when this happened, but the city of Parchment area was also part of our proposal for the multi-site study award. So we proposed to use two very distinct study sites, the Belmont Rockford area in northern King County and the Parchment and Cooper Township area, which was impacted also by PFAS, also from industrial source, also presumed to be leaking from a landfill and then contaminating water used for drinking water via that route. So in Parchment, the context is a little bit different. Their municipal supply was contaminated, whereas in Belmont Rockford, it's really a private residential well scenario. But the levels in Parchment were very high and were very homogeneous across that city's water supply. So it provided a really, I think, compelling population to study as part of the multi-site study. And that contamination in Parchment occurred was discovered in 2018. So where we are now is the multi-site study has just wrapped up. Like, we were awarded that. We, sort of implemented data collection along with all the other 7 sites in the country. And now, just as of late last year, all of that data collection is wrapped. So we're done. There are over 7,000 people who joined nationwide, and we're now entering that really exciting phase of the study, which is the data analysis phase. And all key analyses are being conducted on the combined nationwide data set. There are some exceptions to that, but in general, the key analyses are really focusing on that nationwide data set. Our first paper out of this is a methods paper, and that's currently under consideration at a journal. So we look forward to publishing that, as well as findings related to the health outcomes that we measured, such as cardiovascular health, thyroid hormones, cholesterol levels, pregnancy outcomes, antibody response to vaccines, and much, much more in the coming months. I'll also note that just like with the North King County exposure assessment, all participants who joined the multi-site study got their blood PFAS test results, as well as their biomarkers of health test results, like everything that we tested. If we could give it back to them, we gave it back to them as part of their participation. So they were able to learn about their blood levels as part of their participation. And I'll just, since we have it next in the icon list here on the slide, I'll mention that MyPES then is the one I want to dive into more, but I'll set the stage for it just a little bit by saying that this was established to be very different from multi-site study in some ways, but very similar in other ways. So this is designed as a longitudinal cohort, but it's in the same impacted communities as the multi-site study. So we're still in Belmont Rockford and Parchment for MyPES, just as we were for the multi-site study. But MyPES actually launched data collection before multi-site study was able to get off the ground. And so MyPES ran its phase 1 of that longitudinal design back in 2020 and 2021, which was during the height of COVID. We popped up again in 2023 for our second phase, and we are now currently in our third phase of MyPES data collection in 2025. I'll just note also quickly that almost 1,000 people so far have joined all 3 phases. So we're really excited to have that robust longitudinal data set. And what I plan to do in just a moment is dive in a little bit to some of the phase 1 results, just a little bit more to give you all a sense of what we found, especially with respect to blood PFAS levels. Okay, but before I dive into MyPES, I want to note 2 other studies that were really an offshoot of MyPES and MSS, and then one that really was not. So the 2 that are kind of offshoots of MyPES are the PFAS exposure and antibody response to COVID-19 vaccine study, which is down here. This is the icon for that. And for this, we had internal state funding to conduct this, and we partnered with researchers at the University of Michigan. This study was conducted during the initial rollout of COVID-19 vaccines. And really what we wanted to do was address this growing literature that had been up until that point showing some really serious associations between PFAS exposure and antibody response to vaccine, but that was primarily in the published literature from child populations. And so we really wanted to leverage the infrastructure that was already in place here for MyPES, like our clinics, our enrolled population who already had PFAS results, et cetera, to really try and examine antibody response to this brand new, like, novel COVID vaccine, and to do so in sort of a novel population, which was that adult population receiving a vaccine for the first time, as opposed to that child vax series, which was more common in the literature at the time. So what we did for this study for PAIR was we invited MyPES participants who were eligible for the COVID vaccine back to our study offices, and we wanted them to come in. So we had them come in both before and then up to 3 time points after they got their COVID vaccine. Serum specimens that they produced during those visits were then tested by enzyme-linked immunosorbent assays, so ELISAs, to measure antibody binding to the SARS-CoV-2 spike and the nucleocapsid proteins. So anti-spike was used as the primary measure of immunogenicity following vaccination, whereas the anti-nucleocapsid response was not expected to follow vaccination and was thus thought to be a good marker of natural infection. So what we found was that neither individual PFAS concentrations nor their evaluation as a mixture were associated with antibody response to the mRNA vaccines that were available at that time that we studied. So we hypothesized that this null finding might have been related to the timing of that vaccination, as these were adults, and perhaps they were past a sensitive window during development, during which the effects of PFAS on this system may manifest. Just a working hypothesis, but those findings were published in the Journal of Exposure Science and Environmental Epidemiology in 2023, and I have a citation for that later at the end of this presentation. And then very, very much like that vaccine study, we were also able to leverage ongoing involvement in our PFAS health studies, and that was from both the multi-site study and MyPES, to understand how people's behavior might change as a result of learning their blood PFAS levels. So this one was funded as a supplement to the multi-site study award, and it's called BALANCE, or Behavioral Adaptability, Learning About Novel Contamination in the Environment. Excuse me. So for this, I was most interested in understanding domains of mental health and healthcare seeking behavior. So for BALANCE, I administered a survey before participants learned their PFAS blood test results. That survey captured stuff like anxiety, depression, and sleep, and it did so using validated instruments. But then I also asked about relevant diagnostic history, and I asked about healthcare seeking behavior and some other questions like that. And then a few months after they got their PFAS test results, I administered a follow-up or a post survey that asked the same people all the same questions, plus extra ones that were specifically about like changes to their behavior based on what they got in terms of PFAS results. Like I asked if their PFAS blood test results were what they expected them to be, sort of subjective perception questions like that. And so we are now finishing up that post-survey data collection. I only have like one small tiny cohort left to finish their post-survey, and then I'll have that data set completed. And I'm really excited about that. We had over 500 people enroll in BALANCE, and I'm hoping all of them that completed the pre-survey will complete the post-survey, and then we'll start diving into those data really soon. So I'm excited about that. And those two were really perfectly timed. Like we were just trying to leverage what was already in place. So they were really like add-ons to multi-site study and MyPES. But Embark, which I have down here in the corner, is totally different. So this project is different from the rest because number one, it's not an offshoot of MyPES or MSS in any meaningful way. And it is not concerned with residents who lived specifically in those impacted areas on the west side of the state that we focused so much attention on. So Embark is funded by the National Center for Birth Defects and Developmental Disabilities, which is in the CDC, and it is concerned with birth defects. And what we do here is we're using leftover newborn dried blood spots to quantify PFAS levels at birth. This was designed as a case control study, so we're recruiting from all over the state. And really, Embark right now is in its really early stages. So we've launched recruitment, it's in data collection, but we're still actively in that phase. So we're actively recruiting and enrolling participants, and I don't have any results yet to share on Embark, but we'll be very excited once we do move into the data analysis phase of Embark. Okay, so all that leaves me with the little bit of a focus I wanted to put towards MyPES to help y'all maybe appreciate the methods a little bit more and some of the findings of that. Okay. So like I said, MyPES started way back in 2020 and it continues today. We are currently in active data collection mode as we speak, and are really looking forward to wrapping it up, you can see here how it fit in with the multi-site study. We used the same study offices, and like you already heard, the same participants were eligible. So in a really meaningful way, if people joined both MyPES and multi-site study, we will have basically an additional data point to use in MyPES through their participation in the multi-site study. And that can help us understand blood levels and how they change over time. And in fact, we even have a greater capacity to do that specific thing because a lot of these people were also part of the original North Kent County exposure assessment, which took their blood even further back in time. And so I have this really nice like time series of within subject changes in blood PFAS levels over time. And I'll show you just a little bit snapshot of what some of those data are looking like. Okay. So to give you a little bit deeper understanding though of MyPES and go through the methods just slightly, I want to point out that our point was sort of generally to understand health effects associated with PFAS. But what we did was we anchored that very general statement to the existing literature in a lot of ways. So we did focus on areas where there was already associations in the PFAS literature. So we focused on immune health, cardiovascular health, kidney function, liver function, thyroid hormones. But there are several other domains of health also covered in our data, either because we collected samples that were tested for different biomarkers, or because we have body measurements, or a whole host of self-reported conditions that were captured via the survey. Okay, so participants that were 12 years and older came to a local study office and completed a pre-scheduled appointment. So during that appointment, they gave blood samples, which we processed on site. We froze and we stored them on site, and then we regularly shipped those specimens out to the health department's Bureau of Laboratories, which is in Lansing. So participants gave us not only venous blood during these visits, which was used for PFAS quantification and for the health biomarker testing, but they also gave us whole blood from a finger poke that was dropped onto a blood spot card. And so that'll be tested for PFAS and will allow us to do hopefully a bunch of cool stuff, including comparing how PFAS partitions to serum compared to whole blood. But participants also completed a lengthy health and exposure survey, and they had body measurements taken, such as height, weight, blood pressure, that sort of stuff. And we also collected a household water sample at the time of their study office visit around that time, just to check their current drinking water levels of PFAS. So this was very intensive for the participants. This was a lot of their time and effort was sort of demanded as part of participation in MyPES. And what we saw was that enrollment was generally high. We weren't quite sure what to expect because we had never launched a health study of this nature before in these areas or anywhere else. And what you can see here are the sort of some of the basic demographics of the folks who joined phase 1 of the multi-site study. So we had more folks join from that Belmont Rockford area that had that North Kent County exposure assessment previously than we did from Parchment Cooper, but not by a ton. You can see that distribution here. And like we saw before, roughly equal number of males and females, but slightly more females joining. And you can see here just like the age ranges and other basic characteristics. So we were encouraged by this participation rate and really sought to retain these people across all the other time points. Although we did allow people to join in MyPES at subsequent phases of data collection if they met the original eligibility criteria. So those ends over time do fluctuate as you would expect. Okay. And now what I want to show you are the blood levels from that population. And again, this is just from phase 1. And what we always like to do is compare it to NHANES. So in this graph, what you'll see is the blue data bars correspond to MyPES participants and the green to NHANES. And so what I've done here is I've rank ordered the PFAS results from most to least commonly found PFAS. And the one thing that you'll notice here is that we are testing for many PFAS. We're actually able to quantify 45 PFAS, including branched and linear isomers of 3 of them. And that's many more than the CDC lab is able to quantify for the NHANES samples. So comparisons aren't always possible. But when they are, you can see that our MyPES people all have certain PFAS in their blood, much like the general population. So look at PFOS, PFOA, and PFHXS here in particular. You can see that our study population had all of our study population had OA and OS in their blood, and many of them had PFHXS as well. Okay, this is actually continued on the next slide. You can see my other point here is that there were a few PFAS for which the enhanced population had greater percent detects than the MyPES population. But these were not ones that were commonly found in the environment in these study areas, and so likely explainable by other sources of exposure. I will add, though, that the average MyPES participant had 15 unique PFAS detected in their blood, and the number of PFAS detected in MyPES participants' blood ranged from 3 to 35. So no one had no PFAS detected in their blood, and the most had 35 unique PFAS. Okay, but that's just detection rates. So what about actual concentrations of PFAS? What you can see here is that when we plot the distribution, and I'm just taking PFOA as an example here, when we plot those concentrations along the x-axis, that MyPES is really shifted to the right, compared to NHANES data. So what I'm showing you here, the lines with the little American flags, those are NHANES data points. And then the circles here, the gold and blue circles, are MyPES population. So that's the mean and 95th percentile from the MyPES population. And what you'll note is how much higher our 95th percentile is compared to NHANES. And if you look at PFOS, which is plotted here, you'll see the same trend. And in fact, we plotted it like this for all of the PFAS that we measured, and those are all available in a technical report that we published after phase 1 came out. And the other thing we highlighted in our technical reports that I'm not belaboring here is that we also saw a difference between sub-study site. So those folks who came from Belmont Rockford compared to the people who came from the Parchment Cooper Township area, their distributions were different. And we could really see a difference based on where they lived in terms of the blood concentrations, especially of PFOA and PFOS. Okay, but another little area that I think is interesting, and so I wanted to highlight here, is what happens when we compare blood PFAS levels for people in that original North Kent County exposure assessment that was back in 2019 to when those same people joined MyPES for phase 1 and gave us blood samples again in 2021. So if you look here at the purple curve and those purple icons, those are showing you the mean and the 95th percentile for a group of people who joined North Kent County exposure assessment in 2018 and 19. And then in blue, you see the data from those exact same people, but in 2020 or 2021, when they came back from MyPES. And we had over 100 people in this category that joined both North Kent and MyPES. And so that's what you're seeing here. Those 100 people are what are plotted here. And what we're able to do is really watch over time how those blood levels change. And you can see that leftward shift with time. So their MyPES levels were lower than their North Kent County levels. So this PFAS is leaving the blood over time. It's doing that slowly, but it is being excreted. And that is exactly what we would expect based on estimated half-lives of elimination for these chemicals. Okay, and so we plot it like that for all of the PFAS, and we saw a similar trend in general. And so, you know, it's exciting to see this. The participants really, really like and have commented about appreciating plotting it this way and looking at it this way, because I think that there is some relief that the PFAS is being excreted and it is leaving. And over time, in general, everyone's levels tend to go down. There are some exceptions, but those are the general trends. And so we really like plotting it this way, and we plan to continue to do this. So we'll plot it like this for people in this subsection who come back for phases 2 and 3. But then we'll also do it for like a separate subset, not just restricted to those people who came to that earliest time point in North Kent County, but like them aside, we'll also be able to do the same kind of plotting just within MyPES and over time in MyPES, and watch blood levels decline over time, hopefully, with participants who come back time and time again for MyPES. And that'll give us a larger N than restricting on those who also joined the early North King County study. And we'll follow them, yeah, like much later in time. Okay. So understanding blood levels is really what we focused on first and foremost for this MyPES data collection during the first phase. But the point of MyPES, as I've already mentioned multiple times, was to understand associated health outcomes. And so we're really now in that phase. Our time point one, phase one data is now in the place where we're doing a much deeper dive into all those health outcomes. And that's really where our sort of head is at right now, especially with respect to that phase one data set. So we're actively writing up and publishing those health effects from MyPES, and our first publication has come out recently. Yeah, I have it here. So it was on thyroid levels. And we have a lot of other information that I didn't cover that was published just in state released reports that cover PFAS blood levels, water levels, a whole bunch of other demographic characteristics that I didn't touch on, and other content. But our plan is to sort of systematically release the health effects findings in the scientific literature and make them freely available. So we always publish them in open access. And yeah, we're looking forward to more of those continuing to come out. Okay. This is the one I mentioned earlier. The antibody response one has also been published and is open access. And we're working through a lot more. So we have several other manuscripts right now that are going through our internal state like clearance process, and also others that are already in external peer review for manuscripts. And that includes topics like serum lipids, we're looking at dyslipidemia, we're looking at body weight and body measurements, and many others as we get to them. Okay, so with that, yeah, I want to wrap up here to leave time for questions or comments. But of course, I want to acknowledge our funding sources, our research team, our participants, and thank you, of course, to all y'all for your attention. And I'll welcome questions.
Thank you very much, Dr. Bailey. Are there questions from the group? Yay, I see clapping hands from Lynn.
So I do have a question.
Thanks, Paul.
The health predicting measurements that you're making, you just referred to that as a group of measurements. Can you give us a little more idea of what is actually being measured? And if this is a sample set that you're going to go back to, meaning do you have them stored for additional questions?
Yeah. Yeah, so we have 45-ish. I don't have the exact list in front of me, but there are 40 something different actual like biomarkers are quantified in their serum samples. So thyroid hormones is like the best example because that one's on my screen right here. Like we measured like TT3, T4, like TSH, all of those. Like a thyroid panel was quantified in their serum samples, a lipid panel, total cholesterol, LDH, HDL, like triglycerides, all of those were quantified in the lipids panel. We have several others like markers of kidney function and of liver function were also quantified. Sort of standard panels, sort of standard like what people tend to get for the most part anyway, if they get like a routine healthcare checkup. That's generally the domain, nothing like really crazy cutting edge in terms of biomarkers. We have some markers of immune function too. But we do not store them for long-term. So to your second question, we don't store them for long-term resampling. But there's a separate program that DHHS is running, which is like a biostorage, like a biobank, basically, that they are intentionally doing that and storing for other purposes. But we really told in our consents for MyPES, we specified what they would be tested for and said that they would not be tested beyond that and that they would be destroyed at the end of MyPES. So that is not a fundamental part of MyPES. The multi-site study, though, and those serum samples are stored centrally at the CDC. They're in the same laboratory as NHANES uses. Those are stored and have some capacity to be tested long-term. They're retained and there are possible, like they can be possibly tested for additional stuff, including metals, so something that the CDC wrote into their consent. So yes and no. For MyPES, pretty much no. For multi-site study, there is a little bit. And then from DHHS perspective more broadly, there is a program to like bank specimens for long-term storage and sort of reanalysis.
Okay, thank you very much.
Yeah, of course. Thank you.
Other questions from the team? I'm not seeing anything. Okay.
Well,
There
I'll
is in
follow
the-
up if I could.
sorry, Paul, just for a second. Alex had asked in the chat. He says
Oh, yeah.
he lived in Parchment in 2022 to 23, and all residents were issued Brita filter pitchers for drinking water during that time. Have you identified the effectiveness of this intervention versus any other interventions during your studies?
Yeah, that's a good question. So as part of these like exposure assessment and health studies, we don't formally try and understand effectiveness of different filtration systems. That's just not an embedded part. But we have looked at those data when we have it incidentally. So we do have some data that has like pre-filter, post-filter quantification of PFAS. And what we see is that especially certified filters, so there's like NSF certification for PFAS on certain filters. Those filters filter out almost all PFAS. I mean, there's high-end limits on that, but we see widespread effectiveness of the granular activated carbon filters, especially the ones that are PFAS certified. And the whole house filters that are also in play in the Belmont Rockford area similarly are tested. Eagle tests those actually pre and post filter to try and understand efficacy and has found that they're very effective as well. But there is a big concern or like relevance to filter maintenance whenever we start talking about filter efficacy, because especially for the activated carbon ones, like they have a lifespan and have to be maintained properly. I'm just going on my memory from what the state did in Parchment. My knowledge is that they provided through the local health departments the NSF certified PFAS reducing filters to folks in Parchment, and that those were granulated activated carbon, but they weren't the Brita style pitcher filters, not for PFAS anyway. Those were under counter mount. What I think the Brita style ones were for was actually for lead, if I'm remembering correctly. So there's a whole different, I think different filters are better for lead than for other contaminants. And if my memory serves, those pitcher style ones might have been for lead, not PFAS, but don't necessarily quote me on that. We would have to go back and look at the, I could look at the response information.
Thank you. That's good answer. Other questions from the team?
Sorry, I turned on my camera and somehow it's sideways. I'm having some computer
Oh, yes,
problems
it
right
is
now,
sideways.
so that's very strange to me, but I'll leave that on for a second. I loved your talk. Love all of the work going on. It's just fantastic. Zeroing in on the thyroid hormones, has anyone linked your PFAS data to the newborn screening thyroid hormone profiles that are run yet?
No, but I want to. And
Okay.
yeah,
I would
go on.
love to. I have all of your vital stats, and I obtained the linked infant mortality file. But I'm about to go after the. I don't know if the fetal deaths were included in the infant mortality file, But my next step was to go to ask for the I was thinking of just the hypothyroidism screening, because the blood spot, I believe, still just gives us only the TSH, and so we'll only have the confirmatory the serum free T4, and that will only be in those that go in for the secondary testing. But yeah, so if there's anything I can do with what I have that is of interest to you, like I know the process for requesting the data, but I don't know if that would be of use for anything you guys are doing.
Yeah, thank you. We should connect after. That'd be great.
I'll send you an email, but
Love
I
it.
loved your talk, just fantastic. So thank you so much.
Thank you. Yeah, wonderful.
So this isn't about the topic that you presented, but could you give us a little overview of your department? Like, how many investigators are there? How big is this? Are you located through the state? Do you encourage participation by students? Is this a career path that our students should be knowledgeable about and interested in pursuing?
Yeah, thank you for asking that question. I love that. All of this whole idea that we're doing like real research is a little bit new. I mean, it really is like these are some of the first projects that are rolling out. So we're housed right now, like organizationally, within a new, it's called Office of Science. And so sits at the bureau level, that Environmental Health Bureau. And in terms of like how many PIs there are, study directors there's only like 2. It's like me and someone else. And then we do have a bunch of support staff, though. We have a bunch of epidemiologists and toxicologists who provide really like just absolutely invaluable like contributions to all of these projects. And they are generally, though, structured organizationally within some other area in the bureau. So we have a lot of epidemiologists who work on routine surveillance, for example. Are just like standard birth effects epidemiologist is the one who I work with on Embark, for example. And so like just super useful to use those sorts of staff that are already in place at DHHS. And so we don't right now have position descriptions or like hire in people as like a full-time just like researcher, like that's not really part of how it's set up. What we hire are epidemiologists and toxicologists and some other subject matter experts like community engagement specialists, things of that nature, who then work collaboratively on these research projects with us as part of the study team. So I think that kind of answers your question in general. I will also note that we are housed centrally out of Lansing, out of the state health department's facilities in Lansing. But the health department is right now still at least partially remote. And so that gives a lot of flexibility for us to be able to recruit folks who aren't actually in Lansing full time. So I like to use that as when I talk to students who are interested in maybe coming this way, I say that, but we don't know what kind of longevity that has. So I don't want to put too much weight in that. And then lastly, to your point, we don't have students really at the moment, although I would love to. So that's something that's under active discussion.
Okay, thank you very much.
Yeah, thank you for that question.
So I think Melissa has left the room.
I drove her off.
More likely somebody came to the door. Okay. Lynn, is there something else we should be doing before wrapping up here?
Well, I think we'll just ask one more time if there are any questions people have lingering. I don't see any in the chat. But this actually concludes our seminar series for the season. So if you are waiting for the next event, that's not going to be until I think September. It's still in the planning phases. So I suppose everyone have a nice summer if we don't see you at other meetings around here. And thank you very much, Dr. Bailey, for contributing to our seminar series and giving us the grand finale for 2024-2025.
Thank you very much.
Thank you so much.
Thank you very much. We really enjoyed your talk.
Thank you.
Bye-bye.
Bye, everyone. Thanks for coming.